The first result set here is looking at extracting phenotype information from de-identified electronic medical records. The default output format is an HTML table and it shows up in your browser tab. But you also have a wide variety of other options for your output format. Here we are looking at document level results since these are de-identified records, but you can definitely view the results at a patient level if you have the patient info within these records. In this result set, we are extracting phenotype information using a combination of HPO ontology as well as a diseases ontology. In addition to the exact phenotypes, we are also extracting a more general term for the phenotype. We can also distinguish whether a phenotype is for the patient or family. Here we've used a family terms ontology to pick up the different terms. In addition to extracting the phenotypes and the family terms, we're also extracting clinical course terms such as acute, chronic, etc. The next column here is extracting clinical modifiers like mild, moderate, left, right, and so on. Note that these two columns are optional, so the phenotypes will still be picked whether or not there is a mention of a clinical course or a clinical modifier. The leftmost columns that are color-coded are specific to this query, and you have full control over the number and order of these columns. The last few columns on the right are what we call as evidence columns, and they show up by default in every single result set. One of the more important aspects of our Lingomatics NLP platform is that we're not just returning a handful of results, but we're also pointing you to where in the documents these results occur. So let's look at an example document. Here you have phrases like has diabetes in his brother and sister. So we've captured the diabetes here, and we've captured the more general term here for both brother and sister. And then you have heart diseases in both sides of the family. So you've captured heart diseases, a more general cardiovascular diseases for the family. And it says he denies any bleeding disorders or bleeding history. So it's negative for bleeding for the patient. And father had brain cancer, so we have brain neoplasms for the phenotype, a more general neoplasms category here for the father. And if you click on the highlighted text, you will be taken to a cached version of the document where you can see all of the results highlighted. This way you can quickly get more context around your results. Now that was looking at de-identified electronic medical records. For pharma, along the lines of what Jane was talking about, we are looking at extracting gene, disease, mutation type information. So the next few results I'll be showing you are from the last 5 years of Medline abstracts. Here we are looking at the mutations associated with a gene. The mutations are picked using a mutations ontology, and the genes are coming from the OntryGene ontology. If we expand on one of these, we can see that all of the mutations are normalized to a preferred term, irrespective of how they are written within the document itself. Normalizing the results this way allows you to cluster results like we see here. Having a structured table of results with normalized values can be very useful for downstream processing. Like I mentioned before, the first two columns here are specific to this query, but then you have the usual evidence columns to the right. Now, if you click on the highlighted text here, you will be taken to a cached copy of the Medline abstract, and all the results for this document should be highlighted within the document as well. Moving on, the next set of results builds on the previous one that we just saw. The results here are presented in what we call a chart format. The main advantage here is that you can use the facets on the left here as filters. Here, in addition to extracting the gene and mutation information, we are also looking at the diseases that these genetic mutations are associated with. For example, if you want to look at a certain gene, you can click on that gene, and then you can notice that the results are getting filtered depending upon your selection. You can also further narrow down your results by clicking on a disease, and then the table gets updated again. In addition, we are also extracting whether the relationship between the disease and genetic mutations are a simple sentence co-occurrence or a direct verbal relationship. So if you click on sentence here, you can see that the disease and the genetic mutations are simply occurring within the same sentence, but don't necessarily have a direct verb connecting them. However, if you click on verbal relationships, you can now see that there is an actual verb that's connecting the disease to the genetic mutations. So if you're looking to do more of an exploratory analysis, then you would want to have high recall, and going with a sentence may be a good option at that point. But if you're looking for more precise results, then you can look for a direct verbal relationship. And the chat format also allows you to export the results as a TSV file. So you can click on the export option here to do that. Then it'll then export a filtered version of your results. And you also have the export column on the far right here. You're able to unselect results before you export, and then you can export the curated results using this export option here. So there's a lot of flexibility within the chart format to view as well as export results. Moving on, here again, we're looking at the last 5 years of Medline. In addition to the gene and mutation information, we are extracting the severity of the phenotype as well as the patient age and last author. For patient age, we've used our age ontology, and just like the mutations and other ontologies, you can see how the different values are normalized here. Greater than 40 years, for example, is written in this format here, and the same holds true for other intervals as well. For the mutation here, we are not only extracting the mutation terms, but we are also able to extract whether it's a general mutation or a point mutation. And we're also able to extract results from text as well as tables. So as long as the information is mentioned within the document, you can even extract other information like patient number, study location, and so on. You have the ability to make any of these fields optional. In addition, you can also add metadata to your results, like we pulled out the last author here. To conclude, I want to note that the platform has a lot of flexibility and can present results in a very easily consumable format. I hope this demo was useful to understand the capabilities of our NLP platform.